Hi, my name is Ping-Chen Kuo from Indiana University School of Medicine. Today I would like to share our study regarding immunoresponsive Jui-Hung modulates the severity of brain injury in cerebral ischemia. LPS is widely used stimulator to trigger inflammatory response. Recently, one study found LPS can trigger anti-inflammatory activity by induction IgG1 expression in macrophage. IgG1 is an enzyme that can convert cis-accotinate from the TCA cycle to itaconate. Itaconate can increase the stability of NERV2 protein, therefore increase NERV2 downstream antioxidant and anti-inflammatory gene expression. Ischemia shows some similar features of LPS. Ischemic injury can also trigger inflammation, but whether ischemia can also induce IgG1 expression is still unknown. In this study, the middle cerebral artery occlusion mouse model was being used to investigate the IgG1 expression after ischemic injury. After ischemic stroke, RNA was isolated from the ischemic brain at different reperfusion time points and around qPCR. The result shows IgG1 was significantly enhanced after reperfusion. Means ischemia can trigger IgG1 expression. We also found IgG1 deficient mice show significantly ischemic brain injury and also show more severe blood-brain barrier breakdown compared with wild type. Microglia is a cell population play a critical role during cerebral ischemic injury. We compare the microglia activation status in both wild type and IgG1 deficient mice. IgG1 deficient mice shows higher microglia activation in the ischemic brain compared to wild type, based on the microglia activation marker CD68 and CD86. Immunostaining also found another microglia activation marker IBA1 was significantly enhanced in the ischemic brain of IgG1 deficient mice. To further identify which cell population expresses IgG1 in the ischemic brain, we use CD11b magnetic beads to separate CD11b positive microglia and the CD11b negative non-microglia cells. After qPCR, we found IgG1 was significantly induced in the CD11b positive microglia in the ischemic brain. And HO1, which is a downstream gene in the NERV2 pathway associated with the anti-inflammatory activity, also been highly increased in the CD11b positive microglia. We also found in the ischemic brain of IgG1 deficient mice, HO1 expression was significantly repressed compared to wild type. Since IgG1 deficient mice cannot provide anti-inflammatory activity through the itaconate NERV2 pathway in the ischemic brain, will it be possible for the exogenous administration of itaconate derivative DMI could rescue IgG1 deficient exacerbate ischemic brain injury? Our results show DMI treated IgG1 deficient mice demonstrate a significant reduction of ischemic brain injury compared to the vehicle group. And HO1 expression in microglia, also significant induction in the DMI treated group. In the conclusion, ischemia can trigger IgG1 expression in microglia and then enhance HO1 expression, which suppresses brain ischemic injury. In the IgG1 deficient mice, loss of IgG1 itaconate pathway shows exacerbate brain ischemic injury. Exogenous administration of DMI can induce HO1 expression in microglia, therefore reduce the brain ischemic injury in IgG1 deficient mice. Our study reveals a novel function of IgG1 in modulating brain injury following ischemic stroke. They suggest targeting IgG1 and or IgG1 mediated signaling pathways may represent a novel therapeutic strategy for the treatment of ischemic stroke.